Hemostasis. It is the prevention of blood loss after vascular injury. Whenever a blood vessel is damaged, the body activates a series of mechanisms to stop bleeding. Hemostasis occurs by two major steps: primary hemostasis and secondary hemostasis. Let us look into each of these in detail, starting with primary hemostasis. It is mediated by platelets and results in the formation of a temporary platelet plug. Following vascular injury, vasoconstriction of the blood vessel occurs due to neural reflexes, endothelin release from damaged endothelium, and platelet mediators. But this alone cannot stop the bleeding. Damaged vessel wall exposes the underlying endothelial collagen. A special protein called von Willebrand factor binds to this exposed collagen. Platelets bind to the von Willebrand factor with the help of GP1B receptors. This is called platelet adhesion. Next step is platelet activation, where platelets are activated by changing their shape. These activated platelets release chemicals like thromboxane A2 and ADP from their granules. More and more platelets are recruited to the site of injury. With the help of GP2B3A receptor, platelets link to each other. This is called platelet aggregation. A temporary platelet plug is formed. It is weak and cannot seal the vessel completely. Deficiency of von Willebrand factor results in a condition called von Willebrand disease. Platelets cannot stick properly to the damaged vessel wall. In a condition called Bernard-Soulier syndrome, the GP1B receptor is defective. This causes defective platelet adhesion. In Glanzman thrombasthenia, there is a defect in the GP2B3A receptor. This results in defective platelet aggregation. Then we move to secondary hemostasis. It is also known as definitive hemostasis. It is mediated by coagulation factors. These are normally present in the plasma in an inactive form. Secondary hemostasis results in clot formation. This occurs in three major steps: formation of prothrombin activator, conversion of prothrombin into thrombin, and conversion of fibrinogen into fibrin. Formation of prothrombin activator. It can occur by extrinsic and intrinsic pathways. Both these pathways eventually merge into a common pathway. The extrinsic pathway begins when damaged tissue releases tissue factor, that is factor 3. Then factor 7 becomes activated to 7A. Factor 10 gets activated to 10A. On the other hand, the intrinsic pathway is activated by factors already present within the blood. The sequence is factor 12, 12A, factor 11, 11A, then factor 9, 9A, factor 8, 8A, and factor 10. The activated factor 10 produced here is also 10A. Hence, we can see that both pathways have converged. Now begins the common pathway where active factor 10, that is 10A, joins with factor 5, calcium, and phospholipids to form prothrombin activator. This converts prothrombin to thrombin. Now thrombin converts fibrinogen to fibrin. Factor 13 strengthens and cross-links them. It gets activated to 13A. This stabilizes fibrin. Bleeding is finally controlled within short time. The clot starts shrinking. This process is called clot retraction. During clot retraction, the platelets contract and excess fluid is squeezed out. This fluid squeezed out is called serum. Clot retraction is an important function of platelets, and that wraps up a high-yield overview of hemostasis.